So now it's my great honor to introduce our second speaker today, Dr. Annie Ciernia. So Dr. Ciernia did her PhD at the University of California, Irvine with Marcelo Wood, studying the epigenetics of long-term memory formation and synaptic plasticity. From there, Dr. Ciernia moved to UC Davis for her postdoctoral work with Dr. Janine LaSalle, working on the neuro and immune systems in the brain with a series of papers examining regulation of chromatin and transcription in the brain and microglia, as well as neurons. So then in 2019, Dr. Ciernia started her independent lab at the University of British Columbia, where she holds a prestigious Tier 2 Canada Research Chair and has established a research program working to understand the epigenetic mechanisms of transcriptional regulation involved in brain plasticity and neurodevelopment disorders. Her lab's current research focuses on understanding how early life perturbations to the epigenome alter microglia neuron interactions in developing brain and lead to lifelong changes in cellular function and behaviors. So I'm very excited to hear her speak today on the epigenetic regulation of microglia immune memory. So please join me in giving a warm, albeit virtual, welcome to today's second speaker, Dr. Annie Ciernia.
Awesome. Thank you so much, Ben, for that lovely introduction. And I'm thrilled to be able to share some work with with all of you that my lab has been doing over the last couple of years. So my lab is located at the University of British Columbia, and we're in this lovely Center for Neuroscience building. And we're really interested in how genes are being regulated in the brain during development and how different cell types turn genes on and off, and how this is important for developing normal function in the brain and also in the context of neurodevelopmental disorders. And so my lab primarily uses mice as our model system. And we are, of course, interested in mice because they represent many of the shared facets with humans. And so this is a little diagram of the mouse and human brain to scale. So obviously there's some differences here in terms of size, but many of the same fundamental biological mechanisms, including neural circuits and how you can link neural circuits and molecules to behavior, are conserved across mammals. And so one of the big advantages in using mice is we can look at their behaviors. So my lab has a whole branch that looks at different types of behaviors related to development. So this is one of our social tasks. We can also look at isolated brain cells from the mice. We can grow those cells in culture or just look at them right out of the brain. And the big advantage is that we can go back and forth. So we can identify specific targets either in the dish and then go back into the mice to test the impacts on behavior, or we can look at things regulated by behavior and then go back and test them in the dish or in isolated brain cells. So this is a really powerful system to try to understand how molecules relate to circuits and cellular function and how that ultimately drives changes in the brain that lead to behavioral adaptation. And so this is probably not something I really need to cover with this group, but my lab is interested in regulation of gene expression, and we look at this at the level of epigenetic regulation, which includes a whole landscape of different mechanisms, including histone tail modifications, DNA methylation, non-coding RNAs, nucleosome positioning, and it's really the combination of all of these different marks that make up the epigenetic landscape that ultimately control gene expression. And we think about epigenetics in the lab as sort of this way by which the environment can provide instruction to the genome to get a different type of phenotypic outcome. And we're interested in this because of this initial developmental origin of disease model that was proposed now almost 100 years ago. And the idea is that it's really this combination of the genes you're born with in the environment that you grew up with that ultimately can impact your risk of disease onset much later in life, across the entire lifespan, in fact. And part of this hypothesis is that this gene bioenvironment interaction is really mediated by epigenetic change under the skin. And so if we could understand what these changes are that are happening in early life that predispose for disease risk, perhaps we could figure out how to modify them or reverse them and potentially come up with some new therapeutic strategies for things like neuropsychiatric disorders and neurodevelopmental disorders. And so I'm going to talk a bit today about epigenetic regulation in one of my favorite cell types, which is microglia, which I'm going to go into in a bit more depth in a second. And I'm going to tell you a bit of a vignette about how microglia regulate gene expression in the brain in the context of brain inflammation. And so what are microglia? So you may not have heard of these cells. They are the resident macrophages or immune cells that live in the brain. And so this is a cross section of the mouse hippocampus, so a piece of mouse brain tissue. And each one of these little golden star shaped cells, each one is a microglia, and they tile nicely across the brain. They make up about 10% of all brain cells. And they're incredibly important for disease and injury. So these are immune cells that are important for responding to invading pathogens, if there's some kind of brain injury. And what's been really cool that's come out in the last 15 to 20 years or so is that microglia actually do a whole bunch more than just fight off disease and the odd infection. And so there's now a whole body of growing work looking at how microglia impact the developing nervous system. And so during development, microglia can interact with the developing neurons in the brain, and they can help regulate how those neurons are connected to each other. And so neurons talk to each other through these structures called synapses, and microglia can actually promote the formation of synapses and also the phagocytosis or engulfment of synapses to have them removed. And this ultimately has what people have shown is some pretty profound effects on how neural circuits are formed during development. Microglia can also impact the number of neurons in the developing brain, so they can engulf live developing neurons and actually eat them. And so that helps control the final number of neurons that make up the brain. They release a whole host of signaling molecules that can talk to other brain cell types. So they can talk to neurons, they can talk to astrocytes, they can talk to the blood-brain barrier, and really influence how all those cells are functioning together in the developing brain. They're responsive to the brain environment. So microglia have a whole bunch of different receptors that allow them to respond not only to immune signals, but also to neurotransmitters, growth factors, and other things that are being released in the developing brain and in the periphery that can cross the blood-brain barrier. So they're really able to help mediate this type of communication across cell types. And of course, we're interested in this because it's cool biology, but also because all of these mechanisms are potentially disrupted in brain disorders, both in the context of neurodevelopment, but also in the context of aging and things like neurodegeneration. And so we wanted to come up with a way to easily try to model what happens to microglia during an inflammatory state. And you can see these inflammatory states in many different brain disorders, but they often develop very slowly. It's not clear if they're a cause or a consequence of the pathology. And we just wanted a very simple, easy model in which we could induce microglial inflammation and then study what happens to the microglia in terms of their genome regulation. And so to do this, we turned to a model system that's been around for a very long time, in which you can inject mice with lipopolysaccharide or LPS. So this is part of the outer wall of the gram-negative bacteria, so something like E. coli. And when you do this, you can give this peripherally, and then it interacts in such a way that in the brain, you can see microglia go from their nice homeostatic ramified state. You give LPS, and within a few hours, you can see them change their cellular morphology. They start releasing a bunch of pro-inflammatory signaling molecules. They turn up pro-inflammatory gene expression, and they show this sort of general immune activated state. And so this is a really nice, easy, reproducible, and importantly, time-locked model in which we can stimulate microglia in vivo and then look to see what happens to gene regulation and an epigenome in response. And so we've been working on this paradigm of microglial memory, and I'll talk a little bit about what that actually means. So in the homeostatic brain under healthy conditions, microglia have sort of basic functions of surveying their local brain environment. If you give some kind of immune response, and this can be a lot of different things that people have looked at. So LPS will work, but you can also look at in utero infections, pesticide exposure, stress, inflammation, diabetes, any of these things that can potentially put stress on the system. And you can see microglia respond. So they show this nice initial immune response. They change their morphology, they release signaling molecules, and that then resolves. If you then give a second stimulation at some later time point, it can be either the same stimulation or a different one, you can get a couple of different responses. So there is one type of response called primed immunity, in which this secondary stimulus, even if it's exactly the same as the first, produces an exaggerated response. So the microglia hyper respond and become super inflammatory, promoting. Sometimes with a different type of secondary response, you can actually get the opposite. So the microglia fail to respond, so they're hypo-responsive, and this is called tolerized immune memory. And this has been well characterized in peripheral macrophage populations, but not very well studied in the context of microglia. And so this time point in between the initial stimulus and the secondary stimulus, we're sort of thinking about this in terms of immune memory. So what is the molecular signal that sort of encodes this information and then modifies the secondary response to produce either priming or tolerance? And our hypothesis is that this is some kind of epigenetic switch between controlling the homeostatic state, and then you get some sort of stimulation that triggers this switch to cause either priming or tolerance up to a subsequent stimulation. And of course, if we could figure out how to control this, we potentially could reverse the problems that happen in the context of chronic inflammation and different types of neurological diseases. So whether it's a priming problem or a tolerance problem, if you understood how this worked, you could switch it back to the homeostatic state. And so we're interested in trying to understand what potentially drives this transition to priming or tolerance. So are there different stimuli that can produce one over the other? And then what happens actually during this memory state that underlies disease susceptibility to a second hit? And this in humans could potentially be years later. And so I'm going to talk about each one of these in turn. So what drives priming versus tolerance? So this has been looked at least a little bit in the literature. And so one of the first papers on this was 2018, in which a low dose of LPS in an Alzheimer's model or in a stroke model produces priming. So you get exacerbated pathology or exacerbated damage. However, if you give 4 repeated low doses of LPS, now you get the opposite. So in the same models, you can drive either priming or tolerance depending on how much LPS the animals got. And then in 2022, also in adults, Zhang showed that a high dose of LPS and then another high dose of LPS a month later produces tolerance. And many people have also started to look at this in the context of development. So this is from the Tremblay lab out at University of Victoria, where they look at maternal high-fat diet and then give LPS in adolescents, and now they see priming. And then Lindsay Hayes did a recent study looking at a viral mimic, so poly-IC, which mimics a viral infection. And she gave an injection during gestation. So this is embryonic day 9.5. And then as adults, she gave them LPS and she sees tolerance. So this is a bit all over the place. There's LPS doses, there's LPS amounts, there's time, there's development. And so this is rather a confusing literature to try to parse what the actual molecular mechanisms might be. And so we thought, okay, let's just try something very simple. What if we just give a little bit of LPS, so a very low dose, or we give a lot of LPS, so a high dose, or vehicle? And then what if we just wait a week? And we've tested this. I'm not going to show you the data today, but both of these doses, all of the inflammation resolves by 7 days. So there's no long-lasting changes that we can detect. And what if we just, when everything's back to normal, what if we give them LPS or vehicle and see what happens? And our hypothesis is that maybe this low dose will produce priming, which would be similar to what other people have seen in the literature, and the high dose will produce tolerance. And so the first thing we did was to look at what actually happens to the animals when we do this. And so we rate their sickness behavior, and this is all done by someone blind to condition. And so we have our normal mice, which are running around the cage, they're grooming, they're sitting up, they're moving. And then we have our sick mice, which are kind of hunched up in the corner. They close their eyes. They look generally pretty unhappy. And in our injection conditions, what I'm showing you is the first injection and then the second injection, spaced by 7 days. The PBS animals don't really show any sickness behavior. So the injections alone, the fact that you're sticking them with a needle doesn't really seem to matter. They're fine. If we give the low dose of LPS, and then a week later we give the vehicle PBS, the animals are completely recovered. So they show no changes in no sickness behavior. If we give the vehicle and then a week later we acutely give LPS, this is like our positive control, we now see nice sickness behavior. And keep in mind, this is a relatively low dose of LPS. And now if we give them the double hit, so low LPS and then a second hit of LPS a week later, we see this nice enhancement of sickness behavior, both in females, which are the pink dots, and males, which are the blue. And that's great. So it looks like something is happening in terms of augmenting the sickness behavior. And if we look now at the tolerance paradigm with the high LPS dose, again, they're back to baseline after 7 days. So this is the high LPS and then PBS. This is the same LPS control condition. And now we've completely blocked the sickness behavior. You actually cannot distinguish these animals that get high and then low from the PBS control animals. They look completely normal. So this was super exciting. This was the first indication that this dosing paradigm really was having some kind of impact, at least at the level of the whole animal. And so what we found is that the low dose enhanced sickness behavior and the high dose blunted sickness behavior. So we then went on to just ask, okay, what's happening to the microglia? And so we can actually isolate the microglia out of the brain using a series of digestion and preparation steps on a Kirkall gradient, and then we can sort them by flow cytometry and look at RT-qPCR. And so what I'm going to show you first is what happens in the low dose priming paradigm to gene expression. So we've looked at a number of different cytokines. I'm only going to show you a couple. And so this is IL-1 beta expression. And this is a pro-inflammatory cytokine that gets turned on in response to LPS. The PBS control animals have pretty low expression. They're basically back to baseline after 7 days after the low LPS. You see a small induction to the single dose of LPS, and you see this whopping enhancement in response to the combination where the first dose is the low and the second dose is LPS. So this looks very much like what we expect from priming. We see a very similar pattern for TNF alpha, which is another pro-inflammatory cytokine. And if instead we look at the tolerance paradigm, now what we see instead of an enhancement is actually a blunting of IL-1 beta expression and a blunting of TNF alpha. So exactly what you would expect for a tolerance paradigm. And so this got us really exciting that the sickness behavior matches the gene expression in the microglia, such that the low dose primes microglial gene expression and the high dose tolerizes it. We also checked to see if this was really being driven by changes in peripheral blood monocytes, which of course are also responding to LPS. I'm not going to show you the data here, but they don't appear to have these memory changes. So they respond to every dose of LPS as if it's the first response. There's no differential changes depending on your prior history. And so we wanted to then ask, okay, so we have these gene expression changes. Are the microglia actually doing anything differently in the brain? And so one of the things microglia do in development and throughout life is that they interact with neurons in the brain, and they can actually phagocytose synapses. And so we've developed a flow cytometry assay to be able to quantify this from the brain. And this gives us a measure of both the number of positive microglia, so how many microglia have eaten synapses, as well as a relative fluorescence intensity for the amount of synapses that they've eaten. So I'm just going to show you the percent positive today. And our hypothesis is that, okay, if we're going to have this priming effect for the low dose, that will enhance phagocytosis, and the high dose will blunt phagocytosis, looking like tolerance. And so this is what we found for the paradigm. So we have our PBS controls, we have our LPS and then PBS samples, we have our acute LPS treatment, and then we have our treatment that got the 2 LPS doses. And we see this nice enhancement in the number of positive, or the percent of positive microglia relative to the normalized to the controls. So we see this increase in phagocytosis with the priming paradigm, exactly as we predicted. In comparison, if we look at the tolerance paradigm, we now have a blunting of this response. So we see fewer positive microglia that are engulfing synapses when we give them the high dose and then the LPS, indicating that we've really blunted or tolerized this response. So this is great. Now we have changes in gene expression, and we have changes in the phagocytosis. So the low enhances microglial phagocytosis and the high blunts it. The next thing that we wanted to do was to ask, okay, so we have these nice changes at 7 days. How long do these effects really last? And can we push this system to really look to see how long lasting these changes are? And so we repeated all of these experiments, except now with a 30-day spacing. In the interest of time, I'm not going to show you the data today, but basically we found the same things. So the low dose produces microglial priming of most of the genes that we've looked at, not all of them. So there are some differences, but we can still see priming of sickness behavior, priming of gene expression, and priming of the phagocytosis response. Similarly, the high dose also produces long-term changes in gene expression, microglial responses, and in sickness behavior, indicating that we're really tapping into some kind of molecular reprogramming event that's lasting at least a month in these animals. We haven't checked longer yet. And so this brings us back to our sort of paradigm of what we're trying to actually test. So do different stimuli drive priming versus tolerance? Yes. So we can robustly produce priming with low dose LPS or tolerance with high dose. So that's great. So what about this immune memory component? What is actually happening molecularly to drive these changes? And so what's actually going on here? What are driving these state changes in disease susceptibility that we are hoping to model with this system? And so we've started to look to see what happens after this first dose of LPS. So now we're just comparing the low dose, the high dose, and the vehicle across time. So there's no second dose in this case. And we're just trying to figure out what are the unique molecular signatures between this low and high dose that might be driving these secondary changes. And so one of the things we're interested in is trying to look to see what happens to the chromatin. And so we actually took this time course data that was recently published in 2020 and reanalyzed it. And one of the experiments they had was they simply gave animals PBS vehicle or LPS, and then they looked at the microglia 12, 24 hours or 7 days later. And they did a tax sequencing. If you're not familiar with this technique, it basically allows you to look at open chromatin, so what parts of the DNA are accessible, and then you can compare across different conditions. And so we compared the PBS controls to each one of these different LPS time points. And we found almost 60,000 differentially accessible regions between these different conditions. And we did some clustering analysis on this in a bunch of detail that I won't talk about today, but we identified this subset of enhancers that looks like it's really responding to the LPS in a stable and persistent manner. And so this is a heat map of the read depth normalized attack signal from this cluster of enhancers. So more signal equals more open. And we identified about 2,000 of these regions that increase their accessibility and then stay that way for at least a week. We haven't looked longer. And we've started calling these sort of like putative memory enhancers that might be mediating some of these changes that we can see in gene expression and phenotype. And so we've started working with a collaborator, David Goslin at the University of Laval, and he's interested also in these LPS-induced microglial changes. And his lab has started to look at this in the context of gene expression and histone acetylation. And so they did an experiment in which they gave animals PBS or LPS, and they used the same high dose that we're using in the lab. And then they just looked a number of time points after this to see what's happening to the transcriptome and epigenome. And David has been very generous in sharing some of his data with us. I'm going to show you a little bit of RNA sequencing and chip sequencing for a histone acetylation mark that marks active enhancers. And so we wanted to know, okay, what happens to the genes that are near our memory enhancers? And so we looked at just those subset of genes across these different time points. And we thought maybe like, okay, maybe these genes just are going to turn on and stay on forever, and that's what will be driving our phenotype. And that's not what we found. And so you can see genes in red that increase expression and then come back down, and they actually return to baseline by 7 days. And then same for genes that decrease expression, they also return back to their baseline levels by 7 days. And so there's not a sustained change, at least that we can see in using this method in gene expression in response to the LPS. We also looked at H3K27 signal over our memory enhancers. And that also returns to baseline by 7 days. So it nicely goes up during the acute inflammatory period and then returns back down to baseline. And so this also doesn't look like the persistent memory signal that we might be looking for to look at in what's actually driving these enhancers to maintain their chromatin accessibility. And so we don't really think that the histone acetylation is really driving these enhancers. Maybe it's driving their formation, but it isn't driving the long-term persistent effect. And so we started to look for what other regulators might make sense to try to pursue. And so we did some DNA motif enrichment analysis within our memory enhancers to look for potential transcription factor binding sites. And we identified this transcription factor PU1, which was the binding sites were significantly enriched within our specific memory enhancers. And then there is one published PU1 ChIP-seq data just from microglia under sort of baseline conditions. And we see even under baseline higher PU1 signal over our specific memory enhancers compared to non-LPS regulated enhancers or to the input samples, indicating that this PU1 transcription factor might be really important for potentially regulating this enhancer dynamics that we're seeing. One of the reasons that we also thought that this might be important was that PU1 is actually a well-characterized pioneer factor, and it's actually required for microglial development. And so it's important for regulating microglial enhancer selection during microglial development. It actually helps open enhancers that are important for specific microglial functions. And so we hypothesized that PU1 might be facilitating this change in chromatin accessibility in response to LPS. And so we've started to look at PU1 in response to our different LPS conditions, and we can do this by a number of different methods. What I'm going to show you right now is some intercellular flow cytometry to measure PU1 levels, protein levels in microglia. And when we look at multiple time points after LPS treatment, in this case, the females are red and the males are green, we can see that there's peak expression that goes up in response to LPS between 24 and 48 hours. And this is slightly offset depending on the sex of the animal. And similarly, if we look at the high LPS dose, we again can see a nice peak in the females. The males are a little bit more confusing. I think we need to repeat this experiment again and see if we can replicate this. But there does look like there are changes in PU1 expression in microglia in response to these high and low LPS conditions. Whether they're not actually differential between this, I think we still need to do some more work on that. And so PU1 transiently increases after both of these. We don't yet know where in the genome this is happening, but that's work that we're going to be doing over the summer. Okay, so our sort of working model for this is that LPS initiates this PU1 dependent opening of chromatin, and that at a subset of these newly opened enhancers, they maintain this accessibility after inflammation and form this sort of memory phenotype. And that this is really what might be driving the difference between priming and tolerance. And so one of the things we've been asking is, how does PU1 find the right place to go? And what's been known in other cell types is that PU1 interacts with the histone acetyltransferase CREB binding protein, or CBP. And CBP is one of the main histone acetyltransferases that adds H3K27 to activate enhancers. And so one of the things we're interested in looking at is CBP and microglia required to actually induce microglial gene expression and maybe target PU1 to some of these specific locations? And so we've been making a conditional mouse model to study this. So I'll just briefly go through what we've been doing. So you can take a CBP floxed mouse, and cross it to a Cre driver line that's only going to express Cre in microglia. So the TMEM119 is unique to microglia. This is a Cre that is fused to the estrogen receptor. And so under normal conditions, the Cre is outside the nucleus and CBP is normally expressed. If you add tamoxifen, the Cre can enter into the nucleus, chomp away on those LOXP sites and produce a deletion. So we can do inducible microglia-specific conditional deletions of CBP. And so it took us a long time to generate these animals, and we're still in the process of validating them, but it looks like we can make nice deletions of CBP. So what's shown here is IBA1, which is a marker for microglia, and then CBP, and you can see we can make these nice specific deletions. And if we quantify this by flow cytometry, we can effectively delete CBP in at least 80% of the microglia. We then looked also at what happens to histone acetylation, and we do see these global changes and increase in H3K27, which is exactly what we would predict. So that increase in H3K27 is lost in the CBP flox mice. So you need CBP there in order to induce this enhancer mark. And so we've started to look at gene expression. So this is very preliminary, but we've looked at IL-1 beta, which is the same pro-inflammatory cytokine that I showed you before. And this responds nicely to LPS. So in the wild type animals, we get a nice increase. In the CBP flox animals, that increase in response to LPS is impaired. And that makes a very nice nifty story in the males. We also did this experiment in females, and we see no effect. So they can turn on their LPS-induced gene expression just fine, at least for this gene, without CBP. And so this is a really cool sex difference. There's lots of literature on sex differences in microglia and regulation. And we're really excited to go in now and do some RNA sequencing on this over the summer to try to understand, is this gene really representative of what's happening, or is this an anomaly? And what's actually going on in these two sexes in response to LPS when you don't have CBP present. And so to wrap things up, what I've showed you today is that low dose LPS is able to produce long-term priming of microglial gene expression and function. And in contrast, high dose produces tolerance. And we think this might be mediated by some interactions between PU1 and CBP that regulates differential enhancer usage and chromatin accessibility. Of course, this leaves lots of open questions. So we want to really know if PU1 and CBP are required for these sort of memory enhancers to form. What's happening to drive these differential responses to the high and low LPS? What kind of sex differences might we expect in this sort of paradigm? And are there other epigenetic marks that dictate priming intolerance that we should be looking at? So we're going to be screening some other things by flow cytometry, but of course that doesn't tell you where they are in the genome. So we're going to be doing with Sheila Tevis's lab some cut and run to actually look at where these things are happening in the genome. And of course, we ultimately wanted to bring this back to something that's maybe a bit more disease relevant. So can we use some of these other second hits, like maybe stress or a disease model, to look at what is actually the ability of these priming intolerance stimuli to actually modulate something in the brain that could be more therapeutically relevant. And with that, I really need to thank the people in my lab who did all the work. So the majority of what I showed you today was done by my PhD student, Jennifer Kim, and my master's student, Morgan Tauris, who are really excellent students, both of them, and they have done a ton of work on this project. And of course, our collaborators and all of the funding that supports this work. And with that, I am happy to take any questions.
Thank you, Annie. That was fantastic. I have some questions, and there are a few in the chat, so I'll start with those. First, we've got two questions from Zainab. I'll ask the first one and then the second after. So they say, neat talk. I'm very intrigued about the priming and tolerance of immune response. Is tolerance directly tied to phagocytosis? So if you were to impact phagocytosis directly, would that reverse the impact? Would that would that reverse tolerance?
Yeah, that's a really great question. So phagocytosis is just one convenient readout of microglia function. It's one of the easiest ones that we can quantify. It's not the only one, however. So the short answer is I don't know, but I would be surprised if it's only phagocytosis. We have looked at microglia morphology, which is sort of a surrogate for what they might be doing in the brain. And we do see morphology changes indicative of priming intolerance. We are going to be looking at other types of functional outputs. You can look at phagocytosis of other types of things. You can look at direct contacts with spines, although that requires a lot of imaging. And the short answer is, I think if you ultimately want to take a big picture view of this, and you wanted to use this therapeutically, and you knew in the disease you were interested in that phagocytosis was the main problem, then maybe that would be a really great approach. So you could identify what genes specifically are related to phagocytosis that are being impacted in the tolerance paradigm, and then target those. And that might be a much more specific way to actually use this as a therapy in the context where you knew you had abnormal phagocytosis, rather than trying to just modulate the entire everything that the microglia is doing, which is what we've done so far.
And then the second question is also wondering if you can identify enhancers that are for both priming and tolerance. And actually then a single enhancer can function as a toggle switch based on what transcription factor binds to them in response to the dose of LPS.
Yeah, that's a really great question. And I would love to know the answer to that. One of the things we have, so the short answer is we don't know yet. One of the things we're planning to do is actually to do attack sequencing from the different LPS doses. So the only data we have so far where we've identified these enhancers is closer to the high dose. So we don't know what happens after the low dose. But that is something we would very much like to know. And I don't know. I would guess there might be a subset of enhancers that would act that way. And I would guess there also will be a number of enhancers that are more specific. But yeah, I don't know. It could go either way. It's a great question.
Right. And then we have a question from Hanukkah Vlaming, who says, that was a great talk. Thank you. I was wondering what is happening to P1 expression? Do you think it is required only for initially opening up enhancers, or do you think it stays around and keeps the enhancers open over time?
Yeah, it's a great question. So we've had a lot of debate about this. We see the protein levels go up, and we're not the only ones to see this. They also go, people have shown this for other types of inflammatory conditions where they go up. In our case, it looks like the increase is transient. So the way we're thinking about it is that they are important for helping remodel these enhancers, but maybe not the maintenance. Although I don't know if you need that increase in order to do maintenance. It depends where things are happening in the genome. And P1 is pretty highly expressed in microglia, even at baseline. And we do see it at baseline at some of these enhancers that we're interested in. So it's possible that these increases are not even required in order to induce this remodeling, that it might be sufficient that you can target it there by some other means, maybe CBP, maybe something else. One of the things that I have written in my current grant that we'll go into review shortly is to actually conditionally delete PU1 in microglia and actually test that to see what happens. You can't delete it in early development because otherwise the microglia die, but you can do it transiently in the adult.
Okay, and then Chad Stein says, really great talk. Sorry if I misunderstood, but it sounded like peripheral monocytes react differently than microglia upon repeated treatment. Do you have any ideas as to why that is?
Yeah, that's a great question. So we started looking at them because we were concerned that what we were seeing in the brain might just be an indirect reflection of something that was actually being driven by the periphery. And so you can imagine a case where like the circulating blood macrophages or monocytes are actually changing their epigenome in some way, and that's what's driving a differential signal into the brain. But that doesn't look like it's what's happening. It looks like it's local changes in the microglia themselves. Why the monocytes don't change is interesting. So they turn over much faster, and it's possible that their sort of epigenetic state is just not going to be able to be as easily maintained because you're replacing them continually from the bone marrow. When people have grown them in culture, you can see some innate immune memory phenotypes, but usually these experiments are only done over the course of like 24 to 48 hours. It's hard to get them to last, in theory, longer than that because their lifespan isn't that long. Microglia, however, are quite unique. In that they're very long-lived. So there have been some cool carbon dating studies done in humans, and the average age of a human microglia is like 4 or 5 years, and some of them are up to like 20 years. So they can live a long time, and they don't get replaced by the bone marrow like really ever, unless there's like a breach of the blood-brain barrier in a disease state. So the microglia replace themselves locally by cell division. And so the microglia that you have in your brain right now are pretty much the microglia that you're stuck with. And so if they get reprogrammed, there's not like, they're not just going to get turned over and replaced by something innate from the periphery.
That's really interesting. So I had a question. I thought the sex difference in the CBP dependence was very cool. And I was wondering if you've looked at expression of the paralog P300 in males and females, and if that could potentially be an explanation.
Yeah, I think we have done it by sex, and I don't think we see any differences. We definitely checked to see if there was compensation in the knockouts, and there's not. So P300 does not increase its expression, and when you knock out CBP, it doesn't mean that it doesn't go to the same spots in the genome. Yeah. We haven't looked at that yet, but it doesn't look like it's compensating, at least in this case. And we did do that experiment in males and females, and I don't think we saw any large sex differences, but I have to go back and check the stats to know for sure. That's an interesting idea, though.
Yeah, and along the line of paralogs, I was wondering, with when you're looking at the primed and the tolerant changes, and you got these changes in attack peak signal, and I know you've looked quite a bit at chromatin remodeling in other projects. Have you looked to see if you get changes in expression of different subunits, since you might have an altered chromatin remodeling complex following LPS stimulation?
That is such a good question. And I really want to get a grant/student to work on chromatin remodeling and microglia, because like almost nothing is known. So yeah, I mean, I do have a postdoc, Megan Rowland, who's working in my lab on chromatin remodeling in neurons in the context of autism and some neurodevelopmental disorders. And I would love to do a parallel set of experiments in microglia. We certainly could go into the RNA-seq that we have and look at the subunits. They didn't come out as like top things, but I don't think that would be expected, because I mean, IL-1 beta is like 200-fold increase. Like there's a bunch of genes at the top that are just crazy in their responses to LPS. And I think we might miss some of the more subtle changes. And I would love to know what like the composition of the BAF complex is in microglia. That is completely unknown.
Yeah, if you find out, let me know. I'd be interested. Okay, and we got one more question from Jackson Hoffman. He says, sorry if you mentioned this before, but is there any difference in how much LPS crosses the blood-brain barrier between the first and second treatment and the high and the low doses?
This is a great question and very controversial in the field. So it's broadly thought that LPS does not actually cross the blood-brain barrier in a significant amount. And there's lots of caveats potentially about that. And the doses we're using it at is below the threshold for like actually causing cell death or like breaching of the blood-brain barrier. And people have tested that a bit. And what I can tell you about the multi-dose is that the Wendelin paper that did a lot of this initial characterization with LPS did not see any changes in blood-brain barrier, even when they gave 4 doses of LPS. We should go in, and it's on our list of things to do, to actually check in our own hands that this is also true, that we're not seeing. So what you can do is actually you can inject a dye into the animal and look to see that it doesn't cross the blood-brain barrier, indicating that the barrier is intact. So it's fairly simple to do, but we haven't done that yet. But given what's known in the literature, I don't think we're having like massive breakdown of the blood-brain barrier in either of these cases. And it's interesting because LPS is used super commonly to activate microglia in a bunch of different contexts. And it's really not 100% clear how the microglia get activated. So the LPS doesn't cross the blood-brain barrier. It likely interacts with the endothelial cells and the blood-brain barrier cells itself, and then they signal into the microglia. So this is a whole area of work that I think would be really cool to look at across like plasticity changes across the blood-brain barrier and lots of things. And the tools are now starting to come out. People have identified marker genes for some of these different cell types that can be used to make transgenic animals. So you can start to do some of these experiments. But yeah, there's a lot we don't know about how that signal actually comes across.
Awesome, thank you.